Hi students, welcome to Ligase Academy. In this section, we are discussing about transcription initiation in eukaryote. In previous video, we are already discussed about different types of RNA polymerases involved in the transcription of eukaryote. Here, we are dealing with transcription using RNA polymerase II. All of you know that transcription is the process of synthesis of RNA from DNA template. Transcription consists of three stages: initiation, elongation, and termination. In previous video, we are already discussed about three different types of RNA polymerase involved in eukaryotic transcription. That are RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. Among them, RNA polymerase 1 involved in the synthesis of ribosomal RNA and RNA polymerase 3 involved in the synthesis of transport RNA and small ribosomal RNA. Here we are discussing about eukaryotic transcription using RNA polymerase 2. RNA polymerase II used for the synthesis of messenger RNA, small interfering RNA, small nuclear RNA, etc. This RNA polymerase II transcription system consists of RNA polymerase II enzyme and 5 general transcription factors. These 5 general transcription factors are transcription factor 2B, A, D, E, F and H. These general transcription factors are required for promoter recognition and initiation of transcription. Following initiation, pole 2 alone is capable of RNA polymerase transcript elongation and proofreading. In addition to general transcription factors, there are additional mediators also associated with this RNA polymerase II transcription system. And these additional mediators enables a response to regulatory factors. And during initiation, the order of general transcription factors binding are transcription factor D, transcription factor A, followed by transcription factor B, then transcription factor F, then E and finally H. Now we are moving to the first stage in eukaryotic transcription, that is initiation. Initiation of gene transcription in eukaryotes occurs in specific steps. These are the steps. First one, RNA polymerase along with the general transcription factors binds to the promoter region of DNA and form a closed complex, that is pre-initiation complex. Then this transition of complex from closed state to open state result in melting and separation of two DNA strands. After the promoter melting, the DNA strand separated. This helps in the positioning of the template strand into the active site of RNA polymerase. RNA polymerase can initiate synthesis of new RNA chain without the need for a primer. Many of these initiated syntheses are aborted before the transcript reaches a significant length, that is approximately length of 10 nucleotides. Once the threshold length of transcript attained, RNA polymerase passes the promoter and then the transcription enters into elongation. Then we are discussing about details of general transcription factors and its role in initiation stage. The first general transcription factor is transcription factor 2D. It is the largest general transcription factor. It consists of many subunits. The main components are TBP and TAF. TBP is otherwise known as Tata binding protein. As the name indicates, this TBP binds to the minor groove of Tata box. Tata box occurs within the promoter region. Promoter region is the region within the DNA where RNA polymerase bind. Then TAF, TBP associated factors. There are 13 TBP associated factors. These are interact with the Tata-less promoters. This transcription factor 2D also contains bromodomains. The second general transcription factor is transcription factor 2A. It is a homodimer, that means it consists of two subunits that are identical to each other. Then it acts as a co-activators. It also associated with the Tata binding protein in transcription factor 2D. Then the next transcription factor 2B. It is a monomer. It consists of only one subunit and they stabilize transcription factor 2D binding. So they have the accessibility to active site of RNA polymerase and they direct the RNA polymerase to start site selection, and binding of transcription factor 2B stabilize RNA DNA hybrid, and this decreases the frequency of abortive initiation during initiation process. Next important transcription factor is transcription factor 2F. They pre-associated with enzyme RNA polymerase and interacting with the transcription factor 2E, they recruit this RNA polymerase to core promoter, and it enhances the affinity of enzyme RNA polymerase to transcription factor 2B. And this is the schematic representation of RNA polymerase along with the transcription factor 2F, and this binds with the transcription factor E and they recruited to core promoter. The next transcription factor is transcription factor 2E. It acts as a platform to recruit transcription factor 2H that involved in the promoter melting. Then comes to the important and final transcription factor 2H. It involved in promoter melting and phosphorylation of serine 5 in carboxy terminal domain of RNA polymerase. It also involved in nucleotide excision repair. It consists of 10 different subunits. Among these subunits, 2 subunits have helicase activity and 3 subunits have kinase activities. The subunits that have helicase activity are XPB, that known as Xeroderma pigmentosum protein B. It helps in promoter opening during transcription because it has 3' 5' helicase activity. And the other one is XPD, that is Xeroderma pigmentosum protein D, and it has 5' 3' helicase activity and it involved in nucleotide excision repair. And the 3 subunits that have kinase activity, among them one of them is KIN28. It involved in the phosphorylation of serine 5 in carboxy terminal domain of RNA polymerase. We will see what happens in stages in initiation. First, it is in a closed chromatin stage, that is, the core promoter in the DNA is in heterochromatinized stage. Then formation of open chromatin mediated by different coactivators. It helps in the assembly of pre-initiation complex. This pre-initiation complex are a multi-protein complex that assembled at core promoter. It consists of general transcription factors and enzyme RNA polymerase. This is what happens in initiation stages. First, these general transcription factors and RNA polymerase bind to the core promoter region of this DNA. These are the general transcription factors. These general transcription factors binding to the core promoter in the order of transcription factor 2D, A, B, F and EH. First, the general transcription factor TF2D binds to the core promoter region. TF2A acts as a co-activators, and then the TF2B that stabilize transcription factor 2D binding to the core promoter. Then the RNA polymerase pre-associated with the transcription factor 2F. This pre-associated complex also interact with the transcription factor 2E, which helps in the recruitment of this complex into the promoter region of DNA. Then the final component, transcription factor 2H, bind to this DNA. This transcription factor 2H function in promoter melting and phosphorylation of carboxy terminal domain. This is what happening in the promoter region during initiation. First, the core promoter consists of a tata box. This tata box binding with the transcription factor 2D along with the transcription factor 2A and B. Then the RNA polymerase enzyme recruited with the help of transcription factor E into this promoter region. Finally, the transcription factor H recruited to this region. After the binding of transcription factor 2H into the promoter region, promoter melting or promoter opening occur. Transcription factor 2E stimulate the helicase activity of transcription factor 2H, and they melt the promoter region in an ATP dependent manner. Then the ribonucleotides enters the active site through secondary channel in RNA polymerase, and RNA synthesis begin and undergo abortive initiation when the size of RNA is about 7 to 14 nucleotide. This picture represents the promoter melting stage after binding with the transcription factor 2H. It melts the promoter region. After the promoter melting, RNA synthesis occurs. Then RNA synthesis in 5' 3' direction. This picture represents the binding of general transcription factors during initiation stage. Next stage is promoter clearance. With the help of transcription factor 2B, RNA polymerase begin the synthesis of RNA beyond 14 nucleotide. When the size of RNA is around 100 nucleotides, RNA polymerase is bound by a negative elongation factor. This negative elongation factor pulls the RNA polymerase and prevent their entry into elongation phase. Also, associated with this complex is a DBP sensitivity inducing factor DCIF. DCIF complex maintains the clamp domain of RPB1 in closed state. It improves the processivity of RNA polymerase. And in this closed state, processing of mRNA occurs, that is capping of mRNA occurs. This picture represents the promoter clearance stage, that is when the RNA polymerase synthesis RNA beyond 40 nucleotide. These factors, negative elongation factors and DCIF, bind to this RNA polymerase and it pulls the RNA polymerase. So capping of RNA occurs in this stage. Then coming to the last stage of initiation, that is promoter escape. As the name indicates, in this stage the initiation complexes escaping from the promoter, and it leads to the elongation phase. After capping of mRNA is complete, the elongation factor called positive transcription elongation factor B will be recruited to this region. This is a kinase that phosphorylate serine residue in DCIF and negative elongation factors and serine 2 in carboxy terminal domain of RNA polymerase. This phosphorylation of negative elongation factors releases pre-initiation complex, that is all general transcription factors except TATA binding protein will dissociate from this region and RNA polymerase move away from the promoter. This is known as promoter escape. After promoter escape, RNA polymerase lead to the elongation phase. This picture gives you a clear image of what happening in promoter escape. The elongation factors, with the help of its kinase activity, phosphorylate DCIF and negative elongation factor as well as serine residues in carboxy terminal domain. And this phosphorylation releases these pre-initiation complexes and the RNA polymerase moves away from the promoter region and enters into elongation phase. That's all about initiation stages in eukaryotic transcription. We will discuss about elongation as well as termination in our next video. Stay tuned to next video. If you like this video, please like, share and subscribe our YouTube channel Lifescience Academy, your educational catalyst.